In the last video, we looked at ATP as a primary source of energy in the cell and how the high energy phosphate bond can be broken to provide kilocalories, i.e. energy, to power various processes within the cell. In this video, we are going to discuss how ATP is actually generated through catabolism. In other words, going from food sources, specifically glucose, and metabolizing that, how can we use that process to generate energy? We will focus this video on three primary processes to process glucose. Those three processes in order are glycolysis, followed by the citric acid cycle, also known as the Krebs cycle, followed by oxidative phosphorylation. Throughout, we are assuming that there is an ample amount of oxygen available to enable full metabolism of glucose under ideal circumstances. Many of these processes take place in the organelle known as the mitochondria, and that's why I have a picture of a large mitochondria here on our entry slide. Before we dig into those processes of catabolism, I want to first lay out a few of the key players in this. Looking at these structures, we see a few that should look familiar and others that will look quite new. ATP, the molecule shown here in the upper right corner, is the structure that we focused on in our last video. And if you haven't yet watched that, it would be a good idea to watch that as a prereq for what we're talking about now. Through hydrolysis, the phosphate bond here breaks from ATP, releasing energy from that very exothermic process and creating ADP as a product of that. This reaction is reversible, so I've drawn the reverse arrow here to indicate that by using energy, we can reform that bond to phosphate. In this video, we focus primarily on the creation of ATP. So we're going to be focusing primarily on this reverse reaction of how we can use energy from glucose to create ATP, and then ATP can later be used to generate energy where it is needed within the body. We also, as a key player in these metabolic processes, talk about acetyl coenzyme A. That's the molecule shown here. It's called acetyl CoA for short. The acetyl part is this 2-carbon group here. This is the part that actually participates directly in the catabolic process. And the CoA group, shown here, this large group, you can really very much think of as the carrier of the acetyl-CoA. So the acetyl-CoA is the portion of this that does the work and participates directly in being converted into ATP and calories, is this acetyl group. The rest of this molecule called coenzyme A is the carrier and is recycled over and over again, carrying various acetyl groups into the process of catabolism. Down at the bottom here, we have a couple of different types of molecules. These two molecules are referred to as FAD and FADH2 as a pair here, and these two are referred to as, on the left, NAD, on the right, NADH. The FADs and the NADs serve pretty analogous roles in oxidation and reduction reactions that are essential during catabolism. These are very, very key players within the generation of ATP. Let's take a look at these structures a bit more closely. We have two different forms of FAD. We refer to these as the oxidized OX form here on the left, which is synonymous with FAD. On the right, we have FAD reduced, or red there written in the subscript, and that can also be referred to as FADH2. Why is this one called FAD and FADH2? Well, if we look at the red portion of the molecule here versus here, we notice that in going from the oxidized form FAD to the reduced form FADH2, we've picked up 2 hydrogens along the way. And that is a general truth in oxidation and reduction reactions, that reduction corresponds to a gain in bonds to hydrogen as one way to define reduction. And so therefore, we can classify this molecule as being reduced relative to the one on the left, because this one on the right has additional hydrogen atoms that have been brought in, meaning that this molecule is reduced, or RAD, relative to the structure on the left that was oxidized. During the process of catabolism, these oxidation reduction reactions of FAD are one of the main mechanisms through which ATP will ultimately be generated. Analogously, NAD, which also comes in two flavors, the oxidized form and the reduced form here on the far right, refers to the fact that in looking at the red portion of the molecule here versus here, note that a hydrogen has been gained in going from here over to here. Hence, this one on the left is the oxidized form. The one on the right is the reduced form. And we can, analogous to our nomenclature of the FADs, we can refer to the one on the left here as NAD and the one on the right as NADH, with the H designating that additional hydrogen that has been gained relative to the starting structure. This reaction, just like the one with FAD, is reversible as we see with the bidirectional arrows, indicating that these molecules can be oxidized and can be reduced. And indeed, that is what will happen during the process of metabolism as we dig deeper into this. Now that we've taken a closer look at these structures, we are ready to dive in to looking at some of the processes that break down glucose to ultimately yield ATP and how these molecules we see here play into that elaborate scheme. Let's take a look now at the process of catabolism, where we start with polysaccharides. Through digestion with the action of acid present in the stomach and a variety of enzymes, polysaccharides are broken down into individual monosaccharides such as glucose. We will proceed using glucose as our example here to illustrate the full process of catabolism, converting glucose into ATP through 3 stages. The first of those stages being the process referred to as glycolysis. The second main stage being referred to as the Krebs Cycle, and finally the third stage being referred to as oxidative phosphorylation. We are going to explore where each of these processes occur, provide an overview of how they occur, and very importantly, look at exactly how much ATP is generated at each of these stages of glycolysis, Krebs cycle, and oxidative phosphorylation to ultimately understand how glucose is such an excellent source of energy for driving processes within the cell. Taking a look first at the process of glycolysis. Glycolysis occurs not within an organelle of the cell, but instead within the cytoplasm of the cell through enzymes that float around within that space. These specialized enzymes catalyze a series of chemical reactions that convert glucose, a 6-atom molecule, into pyruvate, which is a 3-carbon molecule. We can see the equation for this reaction here and the structure of pyruvate. Since glucose has 6 carbon atoms and pyruvate has only 3, to balance this equation, 1 glucose becomes 2 pyruvate molecules. In addition, during this reaction, we also generate ATP, 2 ATPs to be specific, as well as 2 NADH molecules. Remember that NADH was one of those forms of oxidizable and reducible molecules where NADH is the reduced form with that extra hydrogen, making it called NADH. Since one of our major objectives here is computing how much ATP we'll actually generate from glycolysis plus the Krebs cycle plus oxidative phosphorylation, I'm going to help us keep track of the ATP we've generated by highlighting the ATP here in yellow to help us find that later on when we go to tally up exactly how much we've made in total. I'm also going to highlight our NADH because what we'll learn a little bit later on down the road is that NADH can also be used to generate ATP, so we need to keep track of our NADH that we are compiling along the way as well. So with glycolysis now in the bag, let's go ahead and take a look at the next step of the process. The next step of the process we can consider a transition step of gearing up for the Krebs cycle, which is where the pyruvate molecule is converted through a series of enzyme catalyzed reactions into acetyl-CoA. In the case of converting pyruvate into acetyl-CoA, what we see is pyruvate has 3 carbon atoms in total. It has the acetyl group, the carbonyl directly bonded to the methyl group, like we see over here as the business end of acetyl-CoA, and it also has this COO group. What happens in converting pyruvate to acetyl-CoA is that bond between the carbonyl group and the COO is broken to release CO2 gas. That's one reason why respiration results in the release of CO2. And the acetyl group from pyruvate becomes bonded onto the sulfur atom of coenzyme A to give acetyl-CoA. So pyruvate donates the acetyl group to coenzyme A, in other words, to create acetyl-CoA. So in other words, one pyruvate molecule gives one acetyl-CoA. And keeping in mind our ratio from glycolysis that we had one glucose giving two pyruvate, what we can really say here is that when we start with one glucose molecule, we generate two pyruvates, because that was the balanced equation we saw here, and then two pyruvates, since there's a 1-to-1 ratio of pyruvate to acetyl-CoA, two pyruvates then gives two acetyl-CoA molecules in this balanced equation. This leads us to the synopsis net equation here, where two pyruvates, which were generated from one glucose molecule, bond to two coenzyme A's to give two acetyl-CoAs. A byproduct of that reaction is CO2 being generated by breaking the bond with N-pyruvate, hence giving two CO2s from two pyruvates. Another important product of this reaction is that the reaction of two pyruvates to give two acetyl-CoAs will also result in the formation of two NADHs. So I'm going to go to put two NADHs in here, and I'm going to highlight in pink like I did above in the glycolysis process so that we can track back to that later. And remember, we need to be able to track back to that later because we will see in the future that NADH can be used to generate ATP. And now with that, we jump into the Krebs cycle, where the Krebs cycle takes acetyl-CoA, and within the mitochondria, the powerhouse of the cell, acetyl-CoA goes through this cycle where each orange block here represents a different intermediate in the process of catabolism. And as we go through this series, what we would notice is that a variety of these reactions represent oxidation and reduction reactions. We can recognize those because NAD and FAD are partners in oxidation reactions, where at each stage that the organic compound is oxidized, such as isocitrate to alpha-ketoglutarate over here, that's an oxidation process, there has to be a partner that is instead reduced, and that partner is NAD+, being reduced to NADH. And that occurs at a few stages throughout here. We see here, here, and here. In all of those cases, the organic molecule is being oxidized as NAD is being reduced. Likewise, when we go from succinate to fumarate over here in the lower left-hand corner of the Krebs cycle, FAD plus becomes FADH2, and that analogously corresponds to succinate being oxidized as FAD plus is reduced to FADH2. So there's an awful lot of oxidation and reduction going on within the Krebs cycle. I mentioned previously that NADH can ultimately be transformed into use as ATP. And so what I'm going to do as we look through the Krebs cycle in general is tally up how much NADH we generate through this cycle of processes. We have an NAD generated here, here, and here as reaction products. So we can summarize that one turn of the Krebs cycle with one molecule of acetyl-CoA will result in the generation of 3 molecules of NADH. So I'm going to go ahead and fill in here. The net is that we generate 3 NADHs per 1 acetyl-CoA. There are also a couple of other important molecules that we should note here. As we look through this, we also see the formation of FADH2, highlighted in gray here in the lower left corner. FADH2 can also ultimately be converted into ATP analogously to NADH, and so I'm going to put 1 FADH2 as a net product here as well. And then finally, I draw your attention to the lowest portion here, where we have GDP becoming GTP. GTP is a molecule analogous to ATP, and in fact, GTP in a 1-to-1 ratio can become ATP, and that's why this is drawn schematically like so. And so I'm going to also go ahead and just put into our equation here 1 ATP, where that ATP is derived from GTP, a very analogous compound. So that takes care of the main business that is created by the Krebs cycle. When we send 1 acetyl-CoA through the Krebs cycle, we generate 3 NADH, 1 FADH2, and 1 ATP. Now keep in mind, in our actual case scenario here, rather than through 1 glucose molecule being able to send just 1 acetyl-CoA through the Krebs cycle, we can actually send 2 through because of this metabolic process here that takes 1 glucose and ultimately generates 2 acetyl-CoAs. And so therefore, in thinking about from the perspective of how much ATP can glucose generate, we should actually say here 2 acetyl-CoAs will result in the formation of 3 times 2 NADHs, so that would be a total of 6 NADHs. And then taking FADH, 1 times 2, gives us 2 there, and then ATP would get doubled to 2 also. I'm going to highlight these like we have in the past, where ATP that was generated through glycolysis in the upper right corner, we'd highlighted in yellow before, so I'm going to highlight it in yellow down here to keep the tally straight. For NADH, I'm going to go ahead and highlight that in our lovely pink color to keep that color coding in agreement with our earlier stages of catabolism. And then FADH2, I'm going to come up with a fun additional color to highlight that we haven't used so far. I'm going to use light blue here to highlight that. So far, if you're keeping score, it doesn't seem like we've generated all that much ATP thus far. We generated 2 ATPs from glycolysis. We've generated 2 ATPs directly from the Krebs cycle. And now we get into where the real magic happens. That is the process of oxidative phosphorylation. Oxidative, as the name implies, requires oxygen. It also occurs within the mitochondria of the cell and consists of the so-called electron transport chain and chemiosmosis to ultimately generate a tremendous amount of ATP through a variety of oxidative processes. So, what goes into oxidative phosphorylation is NADH and FADH2, and guess what? We have a lot of NADH and FADH2 from these other processes that led up to oxidative phosphorylation. So, assuming that we have ample oxygen in the cell and the cell doesn't need to turn it into anaerobic metabolism, the NADH and FADH2 will passage through oxidative phosphorylation to ultimately be converted through a series of chemical reactions into the generation of ATP. So in other words, these molecules, NADH and FADH2, are going to drive the conversion of ADP into ATP. They're going to drive the attachment of that extra phosphate group in order to enable ATP to exist. So how much ATP are we talking about that we can generate from NADH and FADH2? Well, let's look at the conversion factors. If a single molecule of FADH2 passes through the electron transport chain and chemiosmosis, that will yield 2 ATPs. So every molecule of FADH2 results in the generation of 2 ATPs in perfect metabolism. On the other hand, every 1 molecule of NADH that passes through full oxidative phosphorylation will generate 3 ATP molecules. So looking at how many FADH2s we have coming into oxidative phosphorylation, we saw that the Krebs cycle would generate 2 FADH2s from 2 acetyl-CoAs. Hence, we can plug in the number 2 down here as 2 FADH2s and do a conversion to determine how much ATP that will generate. Setting this up with ATP in the numerator and FADH2 in the denominator, what that allows us to do is cancel off FADH2 to determine exactly how much ATP we will generate from complete oxidative phosphorylation of our 2 FADH2s. And that turns out to be a grand total of 2 times 2, making 4 ATPs that we would generate from oxidative phosphorylation of FADH2. I'll go ahead and highlight our 4 ATPs in yellow to match the color coding we've been using all along to help us keep track of our ATPs. We do a similar process for NADH, keeping in mind that 1 NADH gives us 3 ATPs. Therefore, I'm going to go ahead and tally up how many NADHs we have in total. We had 2 from glycolysis, highlighted in pink, 2 from the conversion of pyruvate to acetyl-CoA, highlighted in pink here, and then 6 from the Krebs cycle, where 2 acetyl-CoAs pass through the full Krebs cycle. So that is 6 plus 4 makes 10 in total. So I'm going to go ahead and fill in down here that we have 10 NADHs entering and completing oxidative phosphorylation. Our ratio here is that we have a total of 3 ATPs that are generated from 1 NADH. So I put NADH as 1 in the denominator and 3 ATPs in the numerator. Cancel off as I can my NADH from the numerator, and the denominator takes 3 ATPs times 10 NADHs in total gives us a total of 30 ATPs there that can be generated from NADH-driven processes in the oxidative phosphorylation. So I'm going to highlight that 30 ATPs there to add that to our tally of ATP. So tallying this all up, we have 2 ATPs that we've generated directly from glycolysis, an anaerobic process occurring in the cytoplasm. Then, within the Krebs cycle and oxidative phosphorylation, We had 2 more ATP directly generated from the Krebs cycle. And then finally, through introducing NADH and FADH2, generated through all these other processes into oxidative phosphorylation, that allowed another 4 ATPs to be generated from FADH2 and 30 ATPs to be generated in total by oxidative phosphorylation of all of the NADH that's created through these processes leading up to here. So adding that all up, we have 30 plus 4 plus 2 plus 2 gives us a grand total of, if all of metabolism is working ideally, 38 ATPs can be generated per single glucose molecule passing through this perfect system of catabolism, beginning with glycolysis, then proceeding to convert pyruvate into acetyl-CoA passing perfectly through the Krebs cycle, and oxidative phosphorylation occurring. I say the word perfect throughout this several times because, depending upon the conditions in which the organism is living, these conditions aren't always ideal, and hence, as a result, catabolism doesn't always yield exactly that ideal of 38 ATPs. For example, if oxygen is limited, then anaerobic metabolism can take over. And an example of anaerobic metabolism is if you've worked out hard in the gym, done a lot of heavy weightlifting, and converted over to anaerobic metabolism, what happens is that pyruvate, rather than being converted into acetyl-CoA, gets converted into some lactate, or also known infamously as lactic acid, which can cause the delayed onset muscle soreness. Analogously, in yeast and some other microorganisms, under anaerobic conditions, pyruvate enters fermentation, and fermentation ultimately generates alcohol, or ethanol to be specific, as the product, and that is how home brews are made. So with this all in mind, the take-home message here is that the catabolism of glucose is an example of how the body is able to very, very efficiently use a small sugar molecule in order to generate lots and lots of ATP. This ATP can then be used to drive chemical processes that would otherwise be endothermic, because ATP, as we learned in the last video, can be hydrolyzed to ADP, and when that happens, it releases a tremendous amount of energy to drive all of the anabolic processes that are necessary for life.